So here in this picture, we see a eukaryote. Okay? We see a eukaryote. How do I know? The first thing you can tell about it is that there is a nucleus. And only eukaryotes have a nucleus. That's like the first thing you can do. Now, once you have identified that this is eukaryote, next question is it a plant or is it an animal? Whenever you see green, it's a plant. Because plants are green for the most part, right? It's a plant. So it's a plant eukaryotic cell. These green parts are called chloroplasts. Okay, they're called chloroplasts. And right now, if you zoom in, they look like coins. Okay, so inside the chloroplast, we're going to have coins. Each coin is called a thylakoid. A thylakoid is the name of each individual coin. Okay, the stack of coins is called the granum. Granum. That's a stack of coins. Okay, let's talk about other organelles here. So of course we're going to be having the nucleus, which is the most important. The reason is it stores the DNA that plant cell got from the parents. And of course the DNA will not just stay there, right? Do nothing. It must make mRNA. mRNA does what? Makes proteins. Thank you. And the proteins are made at the ribosome level. Okay, what else do we see? We see here a huge sac that's full of water, which makes the plant look full. This huge sac is called a vacuole. Only plants have vacuoles, okay, that we don't have basically. So animals don't have vacuoles, plants have vacuoles. It's called a vacuole. We can also call it the central vacuole. Vacuole or central vacuole. Okay, this zigzag shape is called a mitochondria. It's responsible for making ATP. Plants, exactly, the powerhouse of the cell. Plants have it, and also animals have it. Okay? As you guys can see, we're going to be having this cell membrane, also called the plasma membrane, which has to be covered by a cell wall, which we lack. Plants must have a cell wall. Why? Because first of all, they're found outside in the environment without any protection. They don't have any bones. They must have a cell wall instead. Number one. Number two, if you over hydrate the plant, will the plant immediately die? No. It might survive for a few days because of the cell wall. It's not going to explode. But right now, if I dare you to drink 5 liters of water in 10 minutes, your cells will start to explode on the inside, which means you will die. Not to a plant though. The cell wall prevents the explosion from occurring. Okay? So, by the look at it, it's a plant cell because of a cell wall, because of a chloroplast, and because of the vacuole. So, a eukaryote will be including plants, animals, and also humans, right? And eukaryotes have organelles. And those organelles, they have their own membranes. And those membranes will separate them and separate their contents and their function from the other organelles. These membranes are going to be called internal membranes, which will result in compartments. It's like when you have a library at home, you have compartments. You place the books here. You place the, I don't know, your other things, maybe notebooks, maybe pencils. That's called compartments. The word in biology is called compartmentalization. The word compartmentalization is going to be very specific to only eukaryotes. Only eukaryotes have compartmentalization. Prokaryotes don't. Okay? Prokaryotes don't. Now, both eukaryotes and the prokaryotes will have some things in common, such as a cell membrane, such as having a cytoplasm, which this book's called a cytosol. Same thing. They both have a DNA, but simply of a different location, right? For example, our DNA is found in the nucleus. When I say our, I mean the human DNA. But the bacteria's DNA is not found in the nucleus. They don't have a nucleus. They're simply found in a membrane in a region without any membranes. Such a region is called a nucleoid. So right now, if I were you, I would start to make a table to compare prokaryotes to eukaryotes. Let's do it together. So, eukaryotes enjoy the word compartmentalization, which basically the placing of organelles within their own compartments. This is very, very important. Why? Because the organelles have their own membranes. When the organelle has its own membrane, we call it a membrane-bound organelle. Okay, but we don't have that in a prokaryote. We don't. That's number one difference. Number two difference, our DNA is in the nucleus, which is obviously going to be a membrane-bound organelle. Membrane-bound just means that a membrane is surrounding it, but their DNA is in a nucleoid region. That's not very protected because there is no membrane. So it's not going to be so protected ultimately. That's why it's going to be much easier for us to kill them with a simple pill because their DNA is very, very exposed. Now, their DNA being exposed has an advantage in a way. We'll talk about it very soon. So because their DNA is not in the nucleus, so it's in the cytoplasm. It's in the cytoplasm. But ours is outside of the cytoplasm. It's in the nucleus. So now if you want to go from DNA to RNA in a eukaryote, you must do it in the nucleus. You want to go from RNA to a protein in the eukaryote, you must do it in the cytoplasm. But guess what? Because a prokaryote has DNA in the cytoplasm, for them it's going to be one simple process. DNA to RNA to protein. All of the three things are found already in the cytoplasm. That means for them the process will be shorter. It will take them a shorter time to make a protein. So less time to make a protein. Now, I want someone here to tell me what DNA to RNA was, the process called. Who knows? I was going to go with transcription. And how about RNA to protein? Thank you. So translation and transcription in the eukaryote will occur in different places. But in the prokaryote, they occur in the same place. So transcription and translation occur in the same place. And here they occur in different places. Good. Let's see what else we can get. So here's going to be a simple diagram of a prokaryote. When I say a prokaryote, I just mean a bacteria. And as you guys can see, they only have a single chromosome. Just one chromosome, which tends to be circular. Right now, it's not so circular, but in fact, it is circular. And of course, bacteria, just like a plant cell, will have a cell wall to protect them. They have a cell membrane. Every cell has to have a cell membrane. They have a ribosome to make the protein, which looks like dots in the cytoplasm. They have a nucleoid. That's where the DNA is stored in their cytoplasm. And they can also move by flagella. Locomotion means movement, basically. So the cytoplasm is going to be that place where the organelles are suspended. Organelles are found in the cytoplasm. If it's a eukaryote, they're called membrane-bound organelles. If it's a prokaryote, they don't have that name anymore because they don't have any membranes. Okay? So, eukaryotes are going to be larger than prokaryotes, and ultimately, structure determines function, the same thing we mentioned back in the first unit. Okay. So, the plasma membrane has to be selective. It does not let everything in or out without controlling the rate. It will let you in, it will let you out, but at a specific amount. And some things will never go in. Will never go in. Because the plasma membrane is going to be selective. Selective. Okay? And also, only some amount can pass per second, which will bring us to the next topic, which is the ratio of surface area to volume. The ratio of surface area to volume is going to be of great importance in the AP exam. They will always ask you about it. The surface area to volume ratio, the larger, the better. And the only way to get a larger surface area to volume ratio is by having a smaller size of a cell. So in biology, a smaller cell is better. That's why you guys have trillions of cells, because they tend to be small to make them better. Smaller cell, better. So, less size of the cell results in an increase of surface area to volume ratio. And that's going to be a good thing. That's a good thing. This will increase the efficiency of the cell. This makes the cell better. Better at multiple things. What are some things that a cell has to be doing on like a minute by minute basis? It has to always take in O2. It has to always take in food or glucose. It has to always release CO2. It has to release waste. To be able to do those things, it must have a high surface area to volume ratio to do those things, to become more efficient. How will this come up in the exam? Let me show you. You see here, they gave us the equations for the surface area and the volume of a sphere, a rectangular solid, a cylinder, and a cube. Here we have an animal cell. Okay, and as we can see, we have the endoplasmic reticulum, which comes in two parts, either rough or smooth. If it's rough, it's carrying the ribosomes on it. If it's smooth, there are no ribosomes on it. The rough one is used to make proteins. The smooth one is used to make other things like steroid hormones. Okay. Here we have our nucleus. It's going to be the biggest organelle in the cell, with the nucleolus inside, which is the place where the rRNA is made, a type of RNA that's involved in ribosome synthesis. And of course, this nucleus has to be covered up by a membrane called the nuclear envelope, which is going to be a double membrane. If it's a double membrane, that basically means it's made of two bilayers of phospholipids. Two bilayers of phospholipids. Like right now, your cell membrane is made of a single bilayer. This one is made of two bilayers. Okay. And of course, we have the plasma membrane, which is going to be selectively permeable. It selects things to go in or out of the cell. The centrosome is going to be made of a pair of microtubules that are going to be so important during cell division, during mitosis, and also during meiosis. Ribosomes, if you find them here on the surface of this bag, they're used for making proteins that leave the cell. The Golgi apparatus is going to be working very closely with the rough endoplasmic reticulum, taking any protein made by it in itself to modify it and then to ship it either to the cell membrane or to ship it outside of the cell entirely. When things get old inside the cell, like an organelle for example, then lysosomes will take care of it and they will digest it and they will also recycle it. Mitochondria, of course, is responsible for making you lots and lots of ATP. 90% of your ATP is made in the mitochondria. We also have the cytoskeleton because your cells don't have bones on the inside. They must have this skeleton on the inside. It's called a cytoskeleton, which is made of microfilaments and microtubules along with intermediate filaments. Whenever you see projections, the word projections, we're talking about increasing of the surface area, which will always be a good thing because it always means more reactions will take place. So most of your DNA is going to be found in the nucleus. The nucleus has almost all of your DNA, which will then be used to make mRNA, which will then be used to make a protein. And of course, some of your DNA is found in the mitochondria. For a plant, that's going to be mitochondria and the chloroplast. Ribosomes are the organelles where proteins get made. They're made of two things, ribosomal RNAs along with proteins. Now, ribosomes can be found in two places. They can be either found attached to the surface of the rough ER, or they can be found simply floating around in the cytoplasm. They will have different purposes. The ribosome is made of two subunits, large and small. The ribosome of the eukaryotes will have a different shape from that of the prokaryotes. It's going to be larger in the case of eukaryotes. Ribosomes come in two varieties, either free or bound to the surface of the rough ER. If they're free, what they do is they make you proteins that stay within the cell. So proteins can also mean enzymes. For example, glycolysis is a process that breaks down glucose and it requires some enzymes. So if the enzymes that are made are to stay within the cell, those enzymes are made by free ribosomes. But if you will make proteins or enzymes that have to leave the cell, then it's made by bound ribosome. Or maybe they will stay on the cell membrane and act as a receptor, for example, or a channel protein. So free ribosomes versus bound ones will have different functions for you. If we're discussing the rough ER, we must also discuss a vesicle because whatever is made by them has to be going inside a vesicle to be shipped to the Golgi apparatus for modification purposes before it gets shipped in a different vesicle to go either to the cell membrane and stay there, or to go to it and to leave the cell entirely. So a vesicle means one of two things. Either it will remain on the cell membrane or it will leave the cell entirely. Let's now discuss the function of the SmoothER. What it does is it will make lipids, it will break down carbohydrates, it will remove the toxic effects of drugs and poisons. For example, right now, let's say you go home and you have a fever and you take a drug. But right now, if you look at the leaflet of the drug, it will have side effects like nausea and other things, for example. But you might not experience any of them. You know why? Because of the SmoothER. It's doing all the work to take down those side effects. Okay? And this SmoothER will also be making for us steroid hormones along with phospholipids that will go and become part of the cell membrane. And the liver will be rich in the SmoothER because the job of the liver is to clear your body out of drugs and poisons. Okay. Here we have a picture of the RoughER, which whatever it makes has to be packaged inside a transport vesicle, which then has to go to another organelle called the Golgi body or the Golgi apparatus. For example, the glycoproteins are proteins that have a glucose on them or a carbohydrate on them. They're made to be glycoproteins inside the Golgi apparatus. That's called modification of the proteins that come to the Golgi apparatus. Lysosomes are going to be so important, not only for committing suicide for the cell to save the neighboring cells. Not only. It can also be important as part of a process named phagocytosis. In the case of phagocytosis, what happens is some organisms don't have a stomach, so they must rely on the lysosome to fuse with a food vacuole from the environment, and then they will digest the contents of this food vacuole using their hydrolytic enzymes. So in this way, the lysosome will essentially act as a stomach for the organism that doesn't have a stomach. Okay? And of course, in a human cell, we also have phagocytosis, not used for the purpose of digesting food, but for the purpose of killing invading cells. So your immune cells include white blood cells, and they are responsible for clearing out any possible infections, right? For example, when you guys get sick, you almost always get fever. It's your body trying to slow things down inside of you, and also to tell you, take a break, take a rest, go to bed. So you're going to have dizziness, you're going to be tired. That's all self-caused by your own body to tell you it's time to quit sports, quit whatever it is you're doing and just go and relax and sleep so I can do my own job, which is helping you survive this viral infection or bacterial infection. So in the white blood cells, they can engulf the bad cells and they can simply do digestion of those cells on the inside. Okay. And of course, the lysosome is going to be also able to fuse with an organelle that has been damaged and needs to be recycled or to get destroyed. They will also take care of that by fusion. For example, let's say the mitochondria is getting too old. It's time to get a new one. We must first trash the first one. But trashing means recycling in the case of the lysosome. Very good. So the lysosome has to fuse with the damaged organelle to be digested and also recycle its inner components. If I mention vacuole, I am not talking about humans. I'm talking about one of two things. Either a paramecium or a plant cell. Either a paramecium or a plant cell. You see, a plant cell is going to have a food vacuole. It's going to have a central vacuole if it's a plant cell. If it's paramecium, it's going to have a contractile vacuole, which basically means a vacuole that's able to contract and help it save itself from having too much water going inside. Right now, if we say, let's go on a drinking water competition, let's see who drinks the first 5 liters in the first 10 minutes. The winner is going to die because your cells will, whoever dies first wins first. So basically, your cells from the inside will explode. And that's not a good thing, obviously, right? And so some organisms like a paramecium live in the environment where there's a lot of fresh water and water keeps going inside. So if they don't expel it, they will explode as well. An explosion means death for a cell. We don't want that to happen. Okay. A food vacuole basically means when a lysosome fuses with the food particle to digest the food inside. And we already mentioned this contractile vacuole for the paramecium that lives in a freshwater environment to be able to pump the excess water out of the cell so it can live. But why would someone need to always have some water but not too much and too little? Because your body has ions. Right now, if you go for a run, and let's say you start to sweat, and then you drink water, you will start to get dizzy. Because your sweat means you lost water as well as salts. You must therefore drink Powerade or Gatorade, I think. Water with salt, not the best taste, but works, yes. Very good. You must always maintain a certain concentration of ions inside of your body. So plants will have a central vacuole which can accumulate a lot of water. It will enlarge when there is water inside this vacuole. Okay, a plant cell is not going to explode because a plant cell has a cell wall, unlike us. Can someone smart here explain to us the process or track the path from the beginning of the nucleus to the end? Great. So basically, the nucleus has DNA, and the DNA makes RNA, but the RNA is not going to stay. It will leave the nucleus, and it will go all the way to the ribosomes on the rough ER to create a product that gets packaged inside a vesicle, which then has to be taken to the Golgi body. Inside this Golgi body, that protein has to be modified, has to be changed in a certain way to either become a permanent part of the plasma membrane or to permanently leave the cell. For example, the pancreas is responsible for the creation of digestive enzymes. But here's the issue. No digestion takes place in the pancreas. So why does it make them? Because they have to make it all the way to the small intestine. That means they have to leave the pancreas and they have to go to the small intestine. Such enzymes are made by ribosomes on the surface of the rough endoplasmic reticulum. That means they then have to make it to the Golgi apparatus, which will then package the products into a vesicle, which will then fuse with the cell membrane and either keep it on the cell membrane or let the contents out, like it's the case for the digestive enzymes made by the pancreas. The mitochondria is going to be very, very important. That's the place where most of your ATP gets made. And prokaryotes don't have mitochondria. eukaryotes do. It's going to be the site of cellular respiration, which is the site where ATP is made in a large degree. Okay, but mitochondria must have O2. That means right now, if you hold your breath, there's no more mitochondria working for you because it requires O2. You can hold your breath for maximum of a minute for most of you. That means your body can only live without O2 for a minute maximum before you start to go unconscious, and then you might die as a result. Okay, chloroplasts, less than a minute. Chloroplasts are also important to make sugars. They make sugar for the plant, they make sugar for the algae, and of course humans are also going to be able to use that sugar. Humans, animals, right? Very good. Now, mitochondria and chloroplast go way back because they have a long history. They were both at some point their own prokaryotic cell. Yes, they were. So at some point, the world was very simple, only prokaryotes, some large and some small. Some were able to make ATP, and some were able to use sunlight to make glucose. But they were just another prokaryote. Here's how the story goes for a theory called the endosymbiotic theory. So we had mitochondria as its own cell, okay? And we had this bigger prokaryote. Now they were both prokaryotes, one small and one big. And they had no rules. The big ate the small. So now when the big prokaryote saw the smaller mitochondria prokaryote, it engulfed it. The word engulf basically means to gulp it in without teeth. Yes, exactly, without teeth. But now the mitochondria made a deal with this larger prokaryote. It said, please don't kill me. I'll provide you energy forever. And of course, the bigger prokaryote said yes. And then they lived happily ever after. And then at some point, because of evolution, this prokaryote became a eukaryote, which was called a human or an animal, because we have mitochondria. Okay? The same story occurred when it comes to the chloroplast as well. So now, chloroplast, mitochondria, and a bigger prokaryote. A bigger prokaryote. And guess what? The same thing happened, engulfed both of them, and they made a deal. They're like, please don't kill me. I will give you ATP and I will give you glucose. And it said, yes, I don't mind, right? I don't mind. And then over time, evolution happened, and then it became a plant, a eukaryote, because a plant has both chloroplast and mitochondria. Now, this might seem like an unreal story. It's actually real. It's actually now a theory called the endosymbiotic theory. A theory means evidence, just like the theory of evolution. It has evidence to it. So what is the evidence behind this theory, behind this funny story? Well, first of all, both the chloroplast and the mitochondria seem to have two membranes, not one. Why is that? Because they already had one membrane when they were still outside, and as they went inside, they acquired a part of this membrane, and now they are able to have two membranes. So mitochondria and chloroplast each has two membranes, not one. That's number one. Number two, they have their own DNA. So it's not just your nucleus that has DNA. Mitochondria and the chloroplast also have some DNA. One more thing, they have ribosomes. And those ribosomes are going to be the same size as a bacteria's ribosome. You know what that means? That it's going to be smaller than a human's ribosome because they were at some point a prokaryote. So the ribosomes are smaller than your other ribosomes. And also, finally, their DNA is going to be circular, just like a bacteria's DNA, circular. Lots of evidence that backs up this theory, so you must believe in it.
Can you repeat the mitochondria?
Yes.
They each have their own.
So look, that's a mitochondria, and that's the prokaryote. So far, before the engulfing occurred, the mitochondria has one membrane, correct? Now, as it gets engulfed, let's do this as red. Okay, 1 second. Yep, and now when we engulf this, it will acquire a part of it, and now it's going to have this additional membrane. And now it has two membranes. One from the bigger prokaryote, one from itself. Now, the mitochondria has two membranes that surround it, and of course those membranes are made of phospholipid bilayer with embedded proteins. Proteins used to carry out reactions, to act as receptors, all that stuff. The inner membrane of the mitochondria is going to be convoluted, which basically means going to look like this to increase the surface area. Okay. This is called enfolding or a cristae to increase the surface area. Okay. So here is the inner membrane. And here is the outer membrane. And there is a space between the two membranes, which is called the intermembrane space. This is the outer membrane, and this is the inner membrane. And the chemical reactions will occur on the inner membrane. The chemical reactions that will ultimately give you ATP will occur on the inner membrane. The space between those two membranes will have a pH that's less than 7 for the most part. It's going to be acidic.
The inner?
The intermembrane space. And here, this is called the mitochondrial matrix. The matrix of the mitochondria. Any reactions that occur in the mitochondria must have O2 supplied to them. A chloroplast has the job of capturing light energy and using light energy, which is free, to make us glucose or to make us sugar. But of course, to capture this energy, it has to rely on the existence of a green pigment called chlorophyll. To ultimately produce sugar that's going to be useful for the body. The chloroplast, just like the mitochondria, will also have two membranes, will also have a space between them that is called the intermembrane space. It will have interconnected sacs called the thylakoids, where a lot of chemical reactions will be taking place. And if you take a stack of thylakoids, that's called a granum, and they will be swimming in a fluid called the stroma. The stroma is where the chloroplast DNA is and where the chloroplast ribosomes are going to be as well. You see here, the single coin is called a thylakoid, the stack is called the granum, and the fluid that they swim in is called a stroma. Peroxisomes are going to be very important. The word oxidation in biology means to break something down. That's what they do. They break something down. So if your body has any toxins, okay, like alcohol, they will take care of it, okay, in the liver. In the process, they will convert the bad things to H2O2, which itself is still bad. So then you have to convert it to water ultimately, which will then leave your body as urine. That's why if you drink a lot of alcohol, you will go to the bathroom a lot.
Do we need to know the process on how it becomes?
No. Just know that it was H2O2 and becomes water. How? No need. Okay. Cytoskeleton is going to act as a skeleton, but simply inside the cell, not only to support, but also to transport. Motility means transportation. So, the cytoskeleton is responsible for giving mechanical support to the cell and also to maintain the shape of the cell. Very, very important for an animal cell because we have no cell wall. Okay. And here are the different classes of cytoskeleton. Let's start with the microtubules. Their protein is going to be called tubulin, and their job is to maintain the cell shape, the cell movement, the movement of the chromosomes when the cell divides, and also when the organelles move. When it comes to microfilaments, they are important for maintaining the cell shape, to change the cell shape, for contraction of the muscle, and also for cytokinesis, which is the last stage after mitosis, where one cell becomes two cells. In a plant cell, they will be fused, but in the animals, they will be disjointed, which means cut in half. Okay. Microtubules are going to be super important because they have a huge role in cell division, a huge role. So there is an organelle called centrosome, which is important for cell division. Okay, a centrosome is made of centrioles, which will be extending spindle fibers. So fibers that are made of microtubules that will attach to chromosomes to move them around, basically. Yep. And plant cells don't have centrioles. Flagella and cilia, they're both used for mobility or motility. So both cilia and flagella are made of microtubules, both of them. Now, some microorganisms that are called protists, they can move in the water by cilia or flagella. Same goes for the sperm of an animal, okay? And that's what they do, basically movement. Now, time to discuss microfilaments, which have a protein called the actin protein. They're used for muscle contraction and also for cytokinesis, which takes place at the end of mitosis or meiosis, to finally make a cell into two. Plants have cell walls, and their cell wall is made of a material called cellulose, which we took back in the first unit. Okay, it's a polysaccharide that's going to be structural. Structural polysaccharide. Plants have cell walls, animals don't have any cell walls. Cell wall is important for what? To protect the plant cell, to maintain its shape, and to prevent it from taking up too much water. And if it did, it's not going to explode. It's also going to be important to hold the plant up against gravity. What this picture shows us is that the membrane, whether it's a plant or not a plant, is going to be important. For example, here we're going to be discussing the proteins that are embedded in the membrane of a cell. called the cell membrane. It has many proteins that are found on it. Some are going to be used for receiving a signal. They're called receptors. Some are going to have an enzyme attached to them to act as a signaling pathway to take the impact of a signal from the outside of the cell to the inside of the cell. If it's a receptor, it's going to be extending from the outside throughout and then from the inside. Some are going to be used for transportation of water. They're going to be called aquaporins. Some for the transportation of calcium, called calcium channels. And some to pump things from low concentration to high concentration, like a proton pump. So aquaporin, calcium channel, and proton pump will all be used for transportation purposes. All of these are going to be termed membrane proteins because they're found in the cell membrane. And here we have the process of cellular respiration, where the mitochondria is responsible for almost all the work. And essentially on the inner mitochondrial membrane, you have a number of enzymes that are responsible for making the ATP and also responsible for the transferring of the electrons across them. And here we have an example of the thylakoid membrane inside the chloroplast, where again multiple enzymes are going to be involved in making the ATP or in taking up light or in transferring the electrons. So ultimately, membranes will be very, very important, whether it's a cell membrane or mitochondrial membrane or chloroplast thylakoid membrane. Okay, so look guys, here we have a cell membrane. Some things will be moving from high concentration to low concentration without the need of any help. This is called simple diffusion. Okay? And some will go from high to low, from the inside to the outside, again without any help, again called simple diffusion. Okay? Now, if they go from high to low, they don't require any energy. That's called passive transport. They don't require any ATP basically. Now, if they go from high to low, while needing the help of a transport protein, that will be called facilitated diffusion. But still, no ATP. Why? Because high to low. ATP has to be used when going from low to high. Because going from low to high is against nature. It's like a homeless person giving a rich person money. It's against nature. Okay? So you must break ATP down into ADP and P. And now once this transport protein called a pump has P, it might become active or inactive. We don't know. The question is going to be telling you. Now, if we're talking about the transportation of huge things, we need a vesicle. To transport things outside the cell, that's called exocytosis. Into the cell, that's called endocytosis. In both cases, there has to be fusion with the cell membrane. In both cases. So in the case of simple diffusion, we have to go from high concentration to low concentration without spending any energy. That means no ATP is needed. And moving from high to low is called moving down the concentration gradient. And we don't need any help from any membrane proteins. No membrane proteins needed. If it's facilitated, it will have some things in common with this one. So, high to low, yes. No ATP, yes. Down the concentration gradient, yes. But it does need the help of membrane proteins called transport proteins or channels. If it's active transport, it's going to be from low concentration to high concentration. And going from low to high means going against the concentration gradient, which will require ATP. It will. And we have to have the help of a transport protein called a pump. We need the help of a pump to do so. So for simple diffusion, it's going to be essentially for hydrophobic things. But if it's facilitated, it's going to be for hydrophilic substances. So hydrophobic things like CO2, like O2, for example, and hydrophilic things like water, glucose, for example. Okay. If it's active transport, it's called a pump, like sodium potassium pump. The word pump means pumping things from the direction of low to high concentration. Okay. How about ions? Where will they fall in? Ions. Hydrophilic. If we're discussing high to low concentration, of course. Polar amino acids. Here we're going to have also non-polar amino acids. And for water, we need a special channel called aquaporin. So in active transport, you must have ATP hydrolysis to break the ATP to get the energy out of it to basically give the process energy to do things. So here's the picture that summarizes all of them. Diffusion means no help of any transport proteins, high to low. If it's facilitated, high to low with the help of transport proteins. But if it's low to high, that's going to be active transport, which relies on ATP hydrolysis to move things from low concentration to high concentration. So the cell membrane is going to be amphipathic. That basically means going to have a hydrophilic region, which is the heads, and also a hydrophobic region, which is the tails. And the tails have to be facing each other. That will make the interior of the cell membrane hydrophobic. That's why the water is going to be afraid of it. So if water wants to go through the cell membrane, it has to go through a special channel called aquaporin to avoid these hydrophobic interiors, basically. Now, of course, the cell membrane is not going to be just made of phospholipids. It must also have membrane proteins, which have to be also amphipathic, meaning that they will be made of a channel like this, with the outside of it being hydrophobic because it's facing the tails, but the inside of it has to be hydrophilic to allow hydrophilic things through it, basically. If we use the word non-aqueous environment, would that be hydrophilic or hydrophobic? Hydrophobic. It's afraid of the water. And you will also see cholesterol found inside the cell membrane, which is going to be important to make that cell membrane fluid. Plants don't have any cholesterol, animals have cholesterol. So the membranes that are found the proteins that are found in the cell membrane are going to be mostly called transmembrane proteins because they span the entire membrane from the outside, from the inside, and also throughout the membrane. So if this is the cell membrane, if it's a transmembrane protein, it's going to be like this, forming a channel in the process or a receptor. Whenever you see ATP on the drawing, that basically means we have active transport, and we have from low concentration to a high concentration. Okay. Some proteins on the cell membrane will act as enzymes. Some will be used for signal transduction. That means signal cannot enter the cell. It binds to the receptor from the outside, causing a signal on the inside. And some proteins will be used to tell the cells that we are neighbors of this type or the other type. Co-transport is going to be a type of active transport, but not purely active transport. This is when something goes from high concentration to low concentration, and the energy of that thing will now be used to power the movement of something from low concentration to a high concentration. So what's co-transport? First, high to low for one substance, which will then power low to high for the second substance. So it's going to be facilitated first, active second. Let me show you the example. So how do we read this? First of all, you will have H pluses that are in a high concentration outside the cell, and now they want to move back into the cell. That's called high to low, right? And secondly, the energy of the movement from high to low will power up this co-transporter to transport sucrose from low to high. So first of all, it's going to be high to low, and the same membrane protein that's doing the work, that's where the low moves to high. So this membrane protein that's doing the work is called a co-transporter. Now if you come across the word pump, what would you think? Low concentration to high concentration. That's why ATP has to be used in this reaction. That's why we have to use the phrase ATP hydrolysis, because we're going to be breaking ATP down. If something big or bulky moves through the cell membrane, you cannot do that without the help of a vesicle. So a vesicle is used for transporting bulky things across the cell membrane. Now, if you're going to be transporting bulky things outside the cell membrane, we call this exocytosis. But if you will take them inside the cell, that's called endocytosis. Both exocytosis and endocytosis require a vesicle to transport bulky things, both of them. Sometimes bringing bulky things into the cell requires a receptor. We call this receptor-mediated endocytosis, because a ligand, this is a ligand, it has to bind to a receptor to allow this pathway to start working. And then once the ligand and the receptor become one piece, only then you can transport things into the cell. Sometimes a cell wants to drink the contents of the solution that's outside of it. When I say that, I mean liquid contents. This is called pinocytosis. But if the cell wants to engulf a solid particle like food, that's called phagocytosis. Phagocytosis means engulfing a solid particle such as food. Engulfing liquid is called pinocytosis. And engulfing using a receptor is called receptor-mediated endocytosis. This is called water potential. It's the amount of water in a certain area. And if you increase the concentration of the solute, you will lower the water potential. They will always go opposite to each other. And the water will always go from the higher water potential to the lower water potential. And water will always go from the less solute concentration to the more solute concentration. water will always move by osmosis. Water will always move by osmosis from the area of higher water potential to the area of lower water potential, or from the area of less solute concentration to the area of more solute concentration, until we establish equilibrium. So, if I have here a cell and the cell's concentration is 0.4, but the solution's concentration outside the cell is 0.6, the water is going to move from the 0.4 to the 0.6. Given that the solution has more concentration than the cell, we call this solution a hypertonic solution, and as a result, the cell will shrink because the water is leaving it. The cell will shrink in size, the cell will shrivel, essentially. And having less concentration in the cell means more water potential. And having more concentration outside the cell means less water potential, and the water always moves from the higher water potential to the lower water potential. Now, if I take the same cell and I place it in a solution of lower concentration, what's going to happen? The water is going to move from the lesser concentration to the more concentration. Because the solution has less concentration than the cell, the solution is called a hypotonic solution.
And the cell expands.
The cell is going to expand. Now, if it's an animal cell, it might explode. But a plant cell is not going to explode, it will live. Now, having a lower concentration means more water potential. And having more concentration means less water potential. And the water always goes from the more water potential to the less water potential. Now, if you take the same cell and you put it inside a solution whose concentration is the same as the cell, so let's say 0.4 and 0.4, we call this an isotonic solution. That means that the water is going to move in and move out at the same exact rate, and your cell will remain the same size. If you have ever had IV fluid put in your vein, you basically put an isotonic solution in your vein so that your cells don't get harmed. Okay? When you guys buy this water from the market, the bottled one, look at the label. It has all these electrolytes. That's good for you. You want electrolytes. You don't want to drink pure water. It will make your cells expand. You want to drink water with electrolytes. Now, there is no taste because there is barely any, but they do exist to a certain extent. So avoid pure water. Always drink electrolytic water. You must always have electrolytes to make your cells not to expand. And of course, always avoid salt water because that will make your cells shrink. Here we have a U-tube shaped container with a membrane that doesn't allow macromolecules to pass. We have macromolecules here that are suspended in the water. And we also have here, but simply less. So, which area has more concentration? Why? Because we have more macromolecules in the same volume of water as the left side. On the left side, we have the same water but less macromolecules, we have less concentration. And water always goes from the less concentration to the more concentration. So water is going to diffuse, and the level of the water on the left side will drop, and the level of the water on the right side will increase until both sides reach equilibrium. The point is, wherever we have less macromolecules, you must have less water, which is the left side. Wherever you have more macromolecules, you must have more water, which is the right side. So the water potential is given by this equation: Poseidon S plus Poseidon P. Now S for solute, which is something that can dissolve in the water, and P for the pressure. Now, the solute potential is equal to negative ICRT. So C for concentration. So if the concentration of the solute increases, the solute potential becomes negative and the water potential will decrease because I have a negative on the right side.
If the concentration increases, how is it negative?
There's a negative sign by default.
So,
By default.
We're talking about the second formula.
So there's always negative in the equation. So whenever you increase the concentration of the solute, you'll always have a negative solute potential. You will always decrease the water potential. Okay. Let me see if there's anything relevant to you. No, it's all good. Easy for you, right?
Yeah.
Okay. So here we've got an animal cell, and as you guys can see, when the water goes inside, too much water will make it burst, which is called lyse. And if too much water leaves it, it will shrivel or shrink. Too much water going inside means that the solution outside has to be hypotonic, and too much water leaving means solution outside has to be hypertonic. But if it's a plant cell and water goes in, it's not going to explode. It will now earn the name turgid. Your plants must always be hydrated, so to avoid wilting. Once your plant starts to wilt, which basically means that the leaves will start to point down, this is a bad sign. It's a bad sign. That means your plant might die very soon. And of course, if the plant loses water, look at the spacing between the cell membrane and the cell wall. It has increased. So if the plant loses water, you can have some gap between the cell membrane and the cell wall, which is not good. Your plant will die, like very soon. So if the solute potential is negative 0.7 and the pressure potential is 0, what's going to be my water potential? You simply have to add them. So 0 plus negative 0.7 will be negative 0.7. Okay, very good. Now, if my cell has a water potential of negative 0.7 and the solution has a water potential of negative 0.9, where does the water go?
inside.
The water will always go from the higher water potential to the lower.
It's gonna go up.
Don't forget that this is negative numbers. So negative 0.9 is less than negative 0.7. It's the opposite. So water will now leave by going from the higher water potential to the lower water potential, and now the cell is going to lose the water. Let's do more. let's say for example the water potential here was negative 0.8, and here the water potential was negative 7. Which one has the higher water potential? The negative 0.8. This one will be the lower water potential, and the water will leave from the higher water potential to the lower water potential, and that's how it goes.